Good morning. During the past 2 days, we heard about the importance and difficulties of a commercial scale manufacturing of a cell therapy product. And so today, I'd like to introduce our technology and how we're going to actually provide a solution to these challenges for especially allogeneic cell therapy product manufacturing. So our mission is start with improving patient life by providing enabling technology to develop and manufacture this novel therapy product. And the vision, and we provide the best manufacturing platform as well as the technical services to unlock the clinical and commercial manufacturing of those products. So we offer single-use bioreactors, you can show on the left-hand side, and various sizes to manufacture those cells. We also provide technical R&D contract development services. We have our own GLP grade research lab that can actually help to develop scalable process for our client and customers. So the next slide is to summarize, as everybody is very well familiar with, as a regenerative medicine, how the cell therapy product, whether it is a human donor-derived or pluripotent stem cell-derived, and then the various type of application. And number one class is cell replacement therapy. So for example, like the beta islet cell for cure the diabetes, and or the cardiomyocyte for heart disease treatment. And then there are various types of neuron cells, chondrocytes, and others, which basically provide the new function to the patient who lost that particular the cell function. And the second category is also very, growing field is immuno-oncology, and especially we already approved the drug for the autologous CAR T, but there's a lot of effort to moving that to the allogeneic CAR T and CAR NK, so you're using immune cells to treat those the cancer, the patient. Third category has been a while, but there is a tissue engineering and artificial cell, the skins, or some artificial the heart and the 3D printing method as well. So those are also take a time, but it requires a large amount of human cells. There is a very positive progress during several years, especially for allogeneic pluripotent stem cell-derived product development. One of the examples, about 2 years ago, the Vertex announced the first in-human treatment for type 1 diabetes patient by injecting the beta islet cell. And then also ViaCyte, not long after that, and they're using gene-edited beta islet cell derived from the gene-edited pluripotent stem cell. But that was a very limited number of a patient. And then this year, in June, the Vertex also announced that there is a total 6 patients treated. They're showing all positive response that endogenous insulin, as well as eliminating insulin use or reducing the insulin use from those treated patients. And then 2 of them are also last 1 year, is stability. And then the general is very tolerating patient. So this is a very encouraging positive data of allogeneic cell therapy treatment. And then not long after that announcement, also the Vertex and Lonza, they announced that the future manufacturing facility. What that means is this will take a long time investment preparing commercial scale manufacturing. So this is very encouraging, but at the same time, as you know, that the current the clinical study, early stage, the research is based on 2D culture or small scale, tea flask or the spinners and so on, which is not necessarily scalable solution. However, a lot of people think that based on the biotech, monoclonal antibody or therapy protein industry experience assume that the single-use bioreactors or stir-type bioreactors, currently around 23,000 liter stainless steel bioreactors used for monoclonal antibody production. That is an easy assumption. However, the key point here is that the allogeneic cell therapy, human cell manufacturing, is significantly different than the biological process, biotech processes. The reason why the biologics, as you can see on the left-hand side, the cell is the only host, and then after the expressing of interest, protein of interest, you throw away the cell. Whereas the cell therapy is the living cell itself is a product. So need to preserve the viability and then their pluripotency, all the cellular functions. And the other important thing in the bioreactor process performance aspect, the biological process is especially CHO cell or E. coli is a single cells. So the single cell suspension in bioreactor and then the average size of 10 to 50 micron. Whereas human cells, except of course NK and T cells, immune cells, but mostly is anchor-dependent cells. So they require to grow on the surface of a hard matrix, it's called the microcarriers, or they grown as a cell aggregate, which is average between 200 to 400 micron diameters. So the particle size is significantly different to be suspended in the bioreactor. And a larger particle obviously need more power input, agitation speed generally, in order to keep suspended uniformly. And that induced that high agitation speed is induced the high hydrodynamic stress. The fluid dynamic conditions are different, and that could be detrimental to the human cells. So cell therapy manufacturing generally requires the more gentler hydrodynamic conditions than the process used in the biologics manufacturing. That's where this PBS vertical wheel, patented vertical wheel technology come into play as a solution. As you can see is that the wheels is rotating vertically, but it creates three different fluid directions, the radial mixing as well as tangential mixing, and then it resulted in very homogeneous mixing it using very low power input. So that, as you can see in the right-hand side, that there is a particle suspension is a microcarrier, but you can see that how well dispersed inside the vessel and with the very the gentle mixing environment. So data, performance data really indicate that how the cell benefit from this kind of a gentle mixing environment. We've been published during last 2-3 years many peer-reviewed articles. However, this year we also got a couple of interesting publications from our customers, which PBS was not involved. This paper was published in May this year by the CBER at FDA, and they demonstrate using vertical wheel bioreactors to support the pluripotent stem cell grown on their microcarriers or as an aggregate. And their conclusion is that this bioreactor system for larger scale expansion of pluripotent stem cell, demonstrating the cell retained the key characteristics of a differentiation potential as a suspension culture. So although this particular paper is using small scale, but this one is demonstrate the proof of concept. And around the same time this year, also University of Alberta and University of Calgary, they use a suspension culture improved iPSC expansion and pluripotency phenotype, and their the conclusion is that the 100-fold iPSC expansion in 5 days using this vertical wheel bioreactor system, and then they believe this is the largest cell growth reported to the date. And then the cells grown in this bioreactor maintain the pluripotency phenotype, and that will definitely the more efficient scale-up strategy and method for down the line. Again, this is still small scale process. So PBS, we have our own internal research lab, We performed our own the pluripotent stem cell expansion study, and also using the perfusion process, we are able to expand. There's a significant improvement. Generally, 10 million cells per mil is a given concentrations, but we are able to push it up to 4 million cells per mil. But more important thing, if you see the three different colors, red, blue, black here is a 0.1 liter scale PBS mini, or the PBS 3 and the PBS 15. As you can see, the growth profiles are also almost superimposed regardless the scale of bioreactor. So this kind of the data, but again, this is showing the culture growth profile of a pluripotent stem cell. But the pluripotent stem cell, the goal is not just expanding the cell, but the ultimate goal is to differentiate it to your target cells. So there are various target cells behave differently, whether it's a cardiomyocyte, neuron cells, beta islet cells. The sensitivity of those processes in bioreactor is all different. So it's not just one process fit all. The expansion phase may be, but differentiation process has become a big challenge. So one of our customers, the ViaCyte, and what we did is scale up their laboratory scale, the roller bottle process, to 30 liter and 80 liter. And also pluripotent stem cell, during this cell expansion, as I mentioned, they're growing as a cell aggregate. The uniformity of a cell aggregate is very important because that critical contributing factor for the quality of final target cells, yield and quality. So the advantage of a PBS vertical wheel is able to generate the uniform size distribution of a cell aggregate. So differentiation process during the multi-step medium exchange, you can maintain the transformation yield in each step. So the key point here I want to make that based on the size of distribution of aggregate, whether it's a 2-liter roller bottle or 3-liter bioreactors, 80-liter bioreactors, a 4-step differentiation process for the endocrine cells, they are very, very comparable. So it's not only the cell growth, but also differentiation, quality, and efficiency is also scalable in these vertical wheel bioreactors. So this slide is just showing the fraction of our customers and the collaborators who agreed to disclose their names. Especially the left-hand side, mostly pluripotent stem cell related customers, and the right-hand side, more microcarrier, MSC, or chondrocyte, and different type of human cell customers. So a couple of them I want to point out is also we did a lot of academic collaboration research at University of Calgary, University of Lisbon, and the University of Alberta. And then also Corning, and recently we engaged in to evaluate their disposable microcarriers because the existing microcarriers, there are great concerns about the residual particulate contamination use. So dissolvable microcarrier hopefully can eliminate that kind of a concern. And currently, so far, the data is very encouraging. We're looking forward to publish the data in the near future. And then another area is the stem cell technology. We're also partnering with them to use their media and in our bioreactor system to help to scale up the researchers, especially academic and early stage startup companies. So they are distributing our mini-scale bioreactors, which is actually displayed in our table in this conference as well. So with that, another point I would like to mention is our headquarters is located in Southern California, the Camarillo in California, which is neighborhood and right next to the Amgen, which is where I used to work. Many of our employees and the executive teams, they are former Amgen employees. So one of the advantages of that, we are leveraging the experience from biotech industry. We all understand what are the GMP requirements and how important all of those. So that we are very much confident about the quality of our product, what we're delivering. And also, last year, we secured the more than $24 million, the funding, and then we're rapidly growing as we speak. And we have a 64,000 square feet facility where we manufacture and testing everything in-house, because in order to maintain the quality of the GMP, the environment. So we are basically know what are the requirements in this industry, and then we're confident that we can deliver those. So we are ready to help your business need and looking forward to serving your manufacturing needs. Feel free to contact us. We're also actively hiring every aspect of business, manufacturing, sales, various areas. So we're happy to report and then our faster growth, and I'm looking forward to working with all of you. Thank you very much.